Hello everyone, and thank you very much for joining us today. My name is Sophie Gerald, and on behalf of the British Neuroscience Association, I'd like to welcome you to the third in this webinar series, Building Bridges Along the Psychiatric Drug Discovery Pipeline. Today, we are considering the academic perspective of the challenges of drug discovery. This is co-hosted with our partners at the Psychiatry Consortium and the British Pharmacological Society. Whilst online life is still very much part of the new normal, we'd like to note our real appreciation for your taking the time to share some screen time with us today. We also ask that you bear with us should Eunice, Dudley or Franklin interrupt connections. Just before we start, and whilst people are still joining, we have a few housekeeping rules to run over. We'll be using the chat and Q&A function today, so please do feel free to say hello and post comments in the chat. Let us know where you're calling in from. But if you have any specific questions for our speakers, then please do make sure you use the Q&A function for these, and you should see both the chat and the Q&A buttons at the bottom of your screen. There will be a Q&A session after the talks with a chance for our panelists to answer any questions you may have. So please do feel free to post them at any time during the talks, and we'll pick them up at the end. You can also ask questions anonymously if you wish to do so. I'd also like to take this opportunity to remind you that this webinar is being recorded with the permission of our speakers and will be available to watch on demand on the BNA's YouTube channel after the event. And finally, just a reminder to please adhere to the BNA's Code of Conduct for webinars and engage respectfully online. We are absolutely delighted to be working with the Psychiatry Consortium, working together to strengthen the links between psychiatry and neuroscience and highlighting the importance of a collaborative process between industry and academia within psychiatric drug discovery. We really hope that this series will help humanise the research story and provide an opportunity to openly discuss the different stages of the research pipeline from all perspectives. So from people with lived experience of mental health and clinicians to academic researchers and industry scientists. As our friend and extraordinary patient advocate Matt Eagle says, drug discovery is about relationships and human interaction. The more human you can make it, the better. So without further ado, it gives me great pleasure to hand over to Ekta Patel. Ekta is only weeks into her new role as Psychiatric Consortium Partnership Manager at Medicines Discovery Catapult. Ekta is a translational scientist with over 8 years of drug discovery research experience in both academia and industrial roles. Ekta is also a strong advocate of patient involvement throughout the drug discovery process, and we're really delighted to be working with her and continuing our work with Laura Ajram on this. So Ekta, over to you.
Thanks for that, Sophie. So hello everyone, and a warm welcome on behalf of the Psychiatry Consortium. So I'm so glad to be here today alongside the BNA in what is our third part of our 5-part webinar series. So for those of you that don't know about the Psychiatry Consortium, we launched back in 2019 in response to a huge patient unmet need in psychiatry with a primary focus on medicines discovery. So we work by bringing together medical research charities, pharmaceutical companies, academics and SMEs to help drive challenging preclinical research for the identification and validation of novel drug targets. So it's hoped that by pulling together this community and providing funding for early high-risk research, it will help us to deliver a pipeline of new treatments for psychiatric conditions. So alongside project funding, we engage the research community globally, and we provide opportunity for collaborations. This is where our partnership with the BNAs, building bridges between industry and academia, aligns so nicely. So this series of webinars aims to foster collaboration between neuroscientists in industry and academia, giving us perspectives and insights from all angles. So today I'm delighted to share the panel with our esteemed colleagues at the British Pharmacological Society, who will co-chair the discussions around the academic perspective of drug discovery challenges. And with that, I hand over to Frida.
Hello, everyone. I'm Dr. Farida Javid, a reader in pharmacology in the Department of Pharmacy at the University of Huddersfield. I am also a fellow member of BPS. I am co-chairing today's webinar with Dr. Sam Groom. Today, we have two fantastic speakers. Our first speaker is Dr. John Davis. John is CSO for the Centre for Medicines Discovery at the University of Oxford and Director of Business Development for the Alzheimer's Research UK Drug Discovery Alliance. John is a biochemist with a PhD from the University of Cambridge. He carried out his postdoctoral training at the Ludwig Institute in Middlesex branch and an EMBO fellowship at the Salk Institute. John joined SmithKline in 1993 as part of the establishment of a neurology research unit within the company. And following the merger to form GlaxoSmithKline, he led non-clinical pharmacological research departments for pain and neurodegenerative diseases. In 2010, John co-founded Convergence Pharmaceuticals, which was subsequently acquired by Biogen, and has since co-founded a further 3 startup companies. In 2015, he joined the University of Oxford to set up and lead the AR UK Oxford Drug Discovery Institute. The institute has developed a portfolio of early drug discovery programs for Alzheimer's and Parkinson's disease, with an emphasis on genetically validated targets, and has leveraged over 100% of additional funding through industrial partnerships. John has over 25 years of drug discovery expertise from target to phase 2 and has helped steer a dozen drug candidates into development and to 4 positive phase 2 proof of concepts. It is therefore my great pleasure to welcome John to this webinar. Over to you, John.
Thank you very much for the introduction, Faride, and thank you for the invitation to speak to you today. So I shall share my screen. Hopefully everything will work. How does that look? Can somebody confirm that you can see it okay?
Yep, that's good.
Okay, brilliant. So I feel slightly guilty in that this particular webinar is titled An Academic Perspective, but as you've just heard, I've spent rather a lot of time in pharma, and I'm either a gamekeeper turned poacher or a poacher turned gamekeeper, depending on which way you look about it. But I'd like to talk to you about my experiences of conducting drug discovery from an academic perspective, and hopefully my views and those of Robbie's will contrast and complement each other nicely. So the first thing I'd like to point out is that really the scientific problems facing either an academic conducting drug discovery research or a professional drug hunter in pharma are faced with the same problems. And I've picked on these here, and I've also left you with a reference at the bottom, which I think is a really good review style article on the considerations, the major considerations and the approach to viewing them that are being taken by professional drug hunters nowadays. So the first thing is you need to pick the right target. Animal disease models can be misleading. Human genetics and genomics have really come to the fore over the last decade or so. If you don't pick the right target, then you know you're onto a loser. You know, fate has already cast its die. And so this is one of the key things, and it's something that we all struggle with, and it's a major issue in academic research as well as in pharma research. The second thing is target engagement. And believe it or not, when I first started in the industry, actually checking that your drug hit the target was not always carried out, especially if you were after a CNS indication. PET studies were expensive, PET ligands were an additional hurdle. And so sometimes trials were run without acquiring any of this type of data, especially in the human setting. But it is vital to have this data, because if you don't see the clinical benefit you're looking for, you do need to know if it's because you got the right target or not, or rather your drug just failed to hit the target, in which case you haven't really tested the validity of the target. And then the final thing brings us back to the patients and the human side once more, which is using the right patient group to test your drug in, and also designing the trial in the right way. You know, this is a huge area in itself and incredibly important. And it's very easy to be swayed by commercial factors such as the market size of a particular patient group. And sometimes that does need to wait, and a drug should be tried out in a more specific patient population first. AstraZeneca use what they call the 5 Rs: right target, right tissue, right safety, the right patient, and of course the right commercial opportunity. And I do encourage you to look at this reference. It's an excellent review. And I think that every partner, whether you're an academic or a professional in the pharma industry, then you should concern yourself with all of these 5 Rs, because you need to be thinking about them all from the very start. The other thing I'd like to point out is that the ecosystem really has been changing over the last few years. Pharma budgets have become much more constrained. You're all familiar with that graph of the escalating cost of bringing a drug to market. Well, a lot of that cost is in the development path. And this, of course, leads less and less money for research. So Big Pharma has had to focus its spend on development, and that has left a little less money for its own in-house research. I think one of the consequences of this is that alliance teams have become more and more active in terms of going out and sourcing early assets. And farmers had to pull in assets for development by buying up biotech companies or licensing IP from universities or seed companies. And these alliance teams in the pharmaceutical industry have become very adept at this. And they're now very proactive, very active, and in fact are trying to collaborate with academics very early in the sort of drug discovery or even the early research path in the hunt for what's going to make the next novel therapeutic. The other side of the coin, of course, is that grant funding becomes ever harder. Universities are under a lot of financial restraint. QR levels have dropped. It's just more and more competitive. And also a lot of charitable funding and the like is coming in with less overheads. And this makes it very difficult for the universities to cover their bottom lines. So as academics, we've had to bring in funds from other sources, and achieving industrial funding for projects is one of those means. And then the last thing is that because of these difficulties and really the insatiable desire of the populace for new therapies to tackle unmet medical needs, then there is a bit of a gap. And the charities, biotech funding, and also contract research organizations that are diversifying their models, they've all risen to the challenge of filling up this gap where there really is a shortage of early R&D in the drug discovery pipeline. And I'm sure you've all heard of the Valley of Death, which is that sort of jump from the basic research across into and through the early translational phase, where it can be quite difficult to get grant funding. So this is undoubtedly creating opportunities for academic drug discovery. And I think I've just highlighted at the bottom here some of the things that have been happening. So target validation, which, as we mentioned at the beginning, is key. The fact is it's human-centric at the moment now. And of course, in academia, we have very good access to patient materials through clinical colleagues. And this is really giving academics a jump start in terms of identifying new targets and making the most of modern population genetics and omics. That feeds through into the demand for novel therapies, because we are identifying the first clues for perhaps a novel therapeutic target. But we need to discharge the risk of those. If they're brand new and nobody's heard of them, then really we're very uncertain as to whether or not they'll be good targets. But this is all feeding this demand for novel therapies. And of course, things like incubators for pushing them through, philanthropic funding as well, is helping us carry out the studies that are required to discharge the risk around these novel approaches. And then further on from that, there are luckily a very diverse range of funders now out there. Again, from philanthropy, a lot of us are discovering our entrepreneurial side, and there's a wealth of VC money at the moment simply because of the economic cycle. So the bottom line is that there really hasn't been a better time for academic drug discovery. So there are many ways of going about this. And I've just listed sort of 3 blocks here, and the colored crescendo bars give you an idea of what might be difficult in one area or an advantage in another area. So on the left, we've got DIY, which basically means you try and do it all yourself, get as far as possible using grant funding or the funding that's very readily available to you through the academic system, and then spin out. There are disadvantages here. It can take a while to raise all this funding. There's often white space between one grant and the next where postdocs leave, etc. This tends to slow you down. On the other hand, there are some advantages. You retain control. The value and the IP which you're steadily accumulating belongs to you and the higher education institute, the university that you belong to. And therefore, you're likely to create more value before you go to market and raise further funds. The incubator, such as Lab 282 and the Psychiatry Consortium, is sort of in the middle. There's a pot of money here that's been put in by pharma and charities, but you do start to lose a little bit of control. Much of the project might be run outside of your institution, using resources and capability in a CRO, but it's slightly less hard work for an academic. And in fact, some of these incubators are superb mechanisms for lowering the energy barrier for an academic. Gives you the opportunity to be involved in some drug discovery, but to still really focus your heart on the basic research and basic research, biology discoveries. And then on the very right-hand side is the last option here, alliance with pharma. This could be big pharma or biotech. One of the advantages is that you can get quite immediate funding, really. These are shorter applications normally compared to a grant application. Companies can make their decisions very quickly. On the other hand, you are going to lose control quite quickly, especially if the project's very interesting. The farmer will pile in the resources, and in the end, the academic's contribution becomes quite small. And of course, as a recompense for that, they will take most of the value. But there are upsides, which I'll come back to in an example. The point I want to make is none of these routes is either right or wrong. It depends on your motivation, the funding climate at the time, and also the therapeutic approach perhaps that you're taking and how popular that is, what the competition's like. So all of these should be considered if you are thinking of conducting drug discovery and are looking for funds to help resource it. And they've each got their place. Now, perhaps because of my history, I'm a particular fan of industrial alliances. And here's an example of one, where we picked up some biology from the University of Manchester. There were a number of good papers which provided a lot of the target validation. But the bioavailability of the compounds was poor. There'd been previous toxicity, CNS penetration was poor. And in fact, the pharmacodynamics of tackling this pathway in the CNS had barely been touched. So we thought this would be a good target for the Drug Discovery Institute here in Oxford. We collaborated with Manchester. We took an assay which they had been running on cover slips, and we miniaturized it into 384 well plates. And then we applied for a grant through what was at the time called the Phenomics Discovery Initiative, which was a collaboration between the University of Dundee, J&J, and Oxford. We won that award, and so ran a screen on the platform that organization provided. And this gave us a starting point. And we developed some chemistries and explored them. And the confidence that that work yielded essentially meant that Accenture was prepared to come in and collaborate with us on this project. And now, of course, they provide most of the funding for running the project. They, of course, are a very skilled AI-oriented chemistry company. And whereas the university and my team can provide more of the sort of physiological biology. And then to boost this project as it's gone along and grown, we've brought in more resources from the Centre for Medicine's Discovery. And effectively, this means that the company has adopted the target. Now, there are some real pros towards creating industrial alliances like this. I've mentioned the immediate funding. And of course, remember that often company funds come with FEC, which is good news for the department heads. But I think one of the biggest things is that when you partner with a company, they can bring in-kind contributions, such as chemistry or access to really good quality ADME profiling or pharmacokinetic profiling. And actually, this in-kind contribution can far exceed the amount of money it costs to fund any work in the academic institution. So you immediately access a huge amount of resource. It would cost you an arm and a leg if you were to achieve it through raising the funds yourself. You also have a joint project team, and more brains on the matter is very important because you've got the diversity of expertise from the two institutions, and you can benefit from an imaginative contract with some risk sharing, which allows you to have some elements of the excitement of a spin out in this type of collaboration. So as I said, this isn't necessarily the right way for you. It might be too much effort. You might prefer to use an incubator. But this is one of the approaches that we certainly advocate here in Oxford. So how do you create a package that will allow you to attract some of this funding? Either an MRC DPFS type funding or an application to the Psychiatry Consortium, or perhaps an industrial collaborator. Well, the answer is that you do have to build up a package that starts to discharge some of those risks that they're concerned about. So some target validation, sound rationale built on genetics, perhaps a lack of toxicity in transgenic animals, some omic data from human patients, maybe some tie-in with some electronic health records. But also what's important is to start to discharge the risk of being able to prosecute a drug discovery program. And here in the Centre for Medicine's Discovery, we've established a number of groups that help do the latter. And of course, these tools that they make can then be used by biologists to help with the target validation. So we have a biotech group that makes constructs and proteins. Then we construct assays that can be built into a screening cascade. And we try to start some medicinal chemistry and pharmacology. And this really is the remit of the Oxford Drug Discovery Institute, this middle block in here. And if we can, we'll do some proteomics to start on the downstream analysis of pharmacodynamic markers. And all the time we're bringing in academic collaborators with biological expertise and clinical collaborators. And the data packages that this type of internal program produces are ideal for attracting industrial funding or even funding from VCs and the formation of spin-outs. So that's all I thought I would say, but I just finish on a couple of important things. There's often talk about the dark side and the light side between pharma and academic research. This really shouldn't be the case. I would quite happily go back to pharma. I'm also absolutely enjoying conducting drug discovery within the academic setting. Everyone has a role to play. We're all in it together. The hurdles are far too big to have any sort of partisan behavior messing it up. So that's one thing I'd like to say. It's a big team effort. The second thing is that the families really need us to work quickly on these key diseases. My own interests are in dementia and a lot on neurodegeneration, but actually I wish we could do more for the psychiatric elements of diseases like dementia and Alzheimer's disease. And remember that every year, about another 200,000 people get diagnosed with dementia in the UK alone. So there's a huge amount of people waiting for us to make progress out there. And lastly, I'd like to thank our collaborators and also our funders, particularly these two trusts, Mr. and Mrs. James Hardwick family and the G&K Boys charitable trusts that have supported us, and of course, Alzheimer's Research UK. Thank you very much.
Thank you so much, John. Really.
Oh, I can't hear you, Sam. I think you've suddenly gone on to mute.
I've been muted, but I'm unmuted now. Yeah, thank you so much, John. Really insightful, an important message at the end. I'm sure, like me, we all have lots of questions for John and more general questions about this topic. Just a reminder, if you want to put those in the Q&A function rather than the chat, that'd be great. I can see a few trickling in already. And then we can talk about those at the end of this session. But for now, it's my pleasure to introduce our next speaker, Dr. Robbie Drake, who is a senior research associate at the University of Bristol. Robbie's research specialty lies in the neurophysiology of chronic pain. And just last week, he was very recently awarded a Medical Research Foundation Fellowship. So I speak on behalf of us all and congratulating you, Robbie, on that. But today, he's going to be speaking about how building bridges with early career researchers can influence the future of psychiatric drug discovery, hopefully for the better. So Robbie, whenever you're ready, please begin.
Thank you. And let me just share. So hopefully everyone can hear and see me. You can hear I'm just recovering from a cold, so apologies if it's a little bit laboured at times. But firstly, thank you for this invitation. This is kind of new ground for me. I'm at the other end to John, just starting my research career. And I guess through part intentional, part serendipitous events, I've been lucky to already work with some pharma and healthcare and medical device companies. So I'm also involved in research culture and the development of young people's careers. And so I thought this would be a good opportunity to kind of talk about the human side of drug discovery that would complement John's introduction to the kind of more medicinal targeting identification side. So I think forming bridges in early careers it benefits drug discovery in two ways. I think one, it allows time for those relationships to develop and really to get that cross exchange of resource and ideas to develop better translational research, which we know is in need in psychiatry. But then for the researchers themselves, I think it diversifies the influences and the mentors and the networks that they have access to. And then this allows us to create better researchers, whether it be here in the UK. I can see we've got attendees from all across the world, but you know, in your local ecosystem, the buzzword that you hear more and more these days. So in this talk, I want to share some experiences to highlight where I've seen the value. It's probably a little bit biased towards the researcher, but hopefully it will be informative for everybody. So just a very quick run through of my career. I don't want to spend a lot of time on this. I can see we're already short of time a little bit. I want to get to the questions. But I came to Bristol in 2010 to do a PhD in neurophysiology. I was a spinal electrophysiologist, and this gave me great technical and experimental training for fundamental science. But it was clear to me that I needed to change the type of research I was doing if I was going to kind of get at the harder questions facing my field to maybe hopefully make some impact on creating some outputs that would benefit people. And I came really captivated with a whole wealth of neuroimaging data coming out of human patients towards the end of that. And it was clear that there was important changes to the brain and how it communicates in the brains of chronic pain patients, but really didn't have much grasp on the causality, the relationship between those changes and the development of the pain state. And I thought, well, here's an opportunity for someone who works in animals with experimental capability to manipulate the systems to provide some insight into aspects here. So I co-wrote an MRC grant with my then supervisor, Bridget Lum, and that was funded. And I think this was my first step really to working towards industry, where I was trying to bridge between clinical observations and preclinical research, and I was trying to develop tools and assays to model or get at aspects of that human condition. And then, of course, in all these stories, it's about being in the right place at the right time and talking to the right people. And I was lucky to have a conversation with some colleagues at Eli Lilly. We together put together a joint application to an internal funding stream, their research award program that was funded in 2019. And that started my world and my eyes to working with industry. To go back to John's point, I think diversifying funding is a really important topic for young researchers. It's very competitive. It's very difficult to get your hands on money. And if you can be creative in bringing new funds in, it can be advantageous to your career. I think the next major step was just recently, really, during COVID, when research activity was low. Again, luck. I had an email pop into my inbox saying that there was a commercialization in science course at Cambridge at the Judge Business School. I thought, why not? I'm not doing a lot else. And it was an eye-opener for me. It was a short course with some lecturing, sort of business 101. And then the real benefit was the sort of small group work in collaboration with a real startup on a real world commercial problem. And, I guess the biggest impact for me was creating networks and talking to people and realizing that I had skills and ideas that were useful outside of my research. And then, as just mentioned, I was recently awarded a fellowship where I brought some of these ideas together, developing tools, refining models, going after the basic science of new targets. And this is in collaboration with a mental health company as well. So I just want to talk a bit about maybe 3 things that I think have been useful in that trip. I'm jumping ahead of myself. I guess the first thing I thought was, well, maybe I need to convince some of those people why ECRs, why early career researchers. The model typically is there is a lot of talented and successful PIs and professors out there with years of experience, and they're the ones that are usually sought after for their opinions. But I'd push on that slightly. I'd say kind of group these ideas into 2 areas. One is that early career researchers, senior postdocs, are really working on the very cutting edge of research typically. And they're answering the newest questions, the ones that are the highest risk, and they're developing tools and techniques to get there. But at least in my experience, this is somewhat removed from the human experience, from the human disease. And so whilst it's great at tackling fundamental science, if we're thinking about drug discovery, it can very quickly go off on a tangent which is unhelpful. And I think bridging early allows that influence, that human-centric influence from industry, just to bring that back online. And it's important to do this early so that the thought can be put into when deciding on design of experiments or what outputs are going to be important, how those outputs are going to inform equivalent experiments in humans. Are there techniques that can be transferred between the 2 subject matters, humans and animals? The second one is this sort of personnel development, personal development. Typically early career researchers are open to new ideas. They're willing to put themselves into new experiences, take on new skills. move geographically. And so again, having that early interaction gives maximum chance to shape those two individuals to create a relationship that's effective over the long term, that is productive over many years. The last point is a slightly generic one about de-risking exploratory science. If we can provide the resource from industry with the scientific freedom of young researchers, then as John pointed to, that there is a nice model there to get at high risk science fairly cheaply. But that's kind of applied to across that academic industry interaction. So now I will talk about three things that I felt have been particularly helpful, and I will deal with the first one, which is direct funding. I mean, obviously, funding is hard to come by, especially when you're starting out. Having a pot of money that you can apply for as the principal investigator is an incredibly rare thing. It may surprise some of you to know that unless you are faculty funded with a salary covered, you're pretty much ineligible for most of UKRI's funding, and you're left with two options as a young researcher. One is to apply for a fellowship, which are highly competitive and fairly few and far between, and that's the standard route. And more what we're seeing is the second route is that you partner with a faculty member who is a PI, and then you can request your salary as a postdoc position. And that's a perfectly acceptable and good way to get scientific ideas off the ground. It then becomes a little bit more difficult when trying to differentiate yourself from that PI or that lab to show some independence. And that's one thing to take into consideration. Leading into these next 2 points, flexibility, streamlined, the beauty of having industrial funding is that they make the rules, and typically they come through a conversation with members of that organization. It allows you to build a project together that you both like. It has input from the company and from yourself. And it's fairly streamlined. It might be 1 or 2 pages of A4, a number of conversations. It doesn't take too much of your time, which again, the 1 or 2 months with 12, 15,000 words, it's a lot of work to put in with a low success rate. So eligibility, being able to be independent, being able to be flexible, to bridge between potentially other projects is all really useful features. On the other side, there's 2 things to think about. The one is the need to publish. I've only been involved at the sort of discovery end, the fundamental end, and of course, there's less of a problem at this thing, and in fact, it's beneficial for both parties. But a young researcher needs to publish. There's no point bringing me in £500,000 of industrial research money and then not being able to show anything for it, unless you plan to leave academia, of course, and join their ranks. So needing to publish as a young researcher is essential. And then my second one is probably prestige. It's clear to me that whilst it's definitely viewed fondly, people view it fondly, that people are bringing in industry money and making connections, it doesn't carry the same kind of weight for an institution as bringing in a fellowship. And so that's something else. Researcher. So a quick look across the internet. These are 3 very quickly found on their websites, as John said, that there are mechanisms and processes to form academic alliances with these companies. They don't specifically mention ECRs, but there's no reason, if those conversations are good and the project's right, that it wouldn't be possible. 2 things that are barriers to this, very much contracts. I'm sure anyone that's been involved in one of these partnerships knows contracts are painful. I do think, again, in my own experience, at this discovery end, at this fundamental science end, it shouldn't be too laborious. It should be easy to pass out foreground and background IP and come up with some standard terms that make sense. My experience is that this was told to be limited to 60 days of negotiations, and it took just over a year. And I think that was unnecessary. The second one is overheads. Universities, rightly so, they lift their costs when they're doing work with industry. No problem with that. I think when we're talking about funding ECR work, if the experimental costs go up and the overheads go up and it's a cash limited fund, then what must come down is a salary. And that's difficult to swallow. I've had to take a salary cut to get my project. I think that could be changed if this was something that was going to be more widely adopted. The next area, I think, one of the problems, I think, for neuroscientists, these fundamental neuroscientists like myself, is that you don't really identify with translation, with drug discovery. My research is not relevant here. I don't play around with molecules. Why would I partner with industry? And I would say pause for thought. I think methods, exchange, and animal model or experimental model development is definitely a ground which gives lots of scope for ECRs to interact. It's where I've had success. So typically, as I mentioned, ECRs are developing sort of state-of-the-art technologies, ideas. They're bringing them to bear on new models and assays, or going back to look at old assays and seeing if they can get more information out of them. But they're high risk. They're really addressing the basic science and having that influence from industry, putting that human condition at the center, getting the ECR to think about, okay, well, if you're developing this model, how will it translate through to clinical studies? How will it be embedded into high throughput screenings or used in an industry? Question pop up earlier, but I think animal behavior is definitely the hot thing at the moment in neuroscience. We're going back to look at animal behavior in a lot more depth, thinking about it in a lot more detail, and this has largely been pushed by the advance or the development of new computer-assisted tracking tools, or AI and deep learning. So Deep Lab Cup is a package of software that's come out from Matthias Lab, and they've been able to now track animals without physical markers in complex systems with multiple groups. And that's really opened up a whole world of new ways to monitor animals in their own environments, doing more naturalistic behaviors with ease. These are open source, so anyone like myself who's not regard myself as a computer user in that way can get access to these really powerful tools. So Robert Datter's lab has used that tracking in combination with AI and deep learning to pass out animal behavior into a sequence of behavioral syllables, and then shown that this is modified by experience, by brain manipulations, by drugs. And then finally, in my own field, looking at the humble mouse paw withdrawal that has been kind of spurned to the edges of research in modern pain. And, they showed that if you sample this paw withdrawal at high rates, use AI, you can pass out aspects of that, features of that paw withdrawal that relate to the reflexive arm, which is perhaps less meaningful in a chronic pain thing. But you can also bring in and identify affective features that are manipulated by high. So the last sort of area to say is it's probably not the hard stuff. The technical stuff is the soft skills, the network building, and this course, Enterprise Tech, really opened my eyes to the fact that the skills required to start a company, to be an entrepreneur, are pretty much the same as those starting a lab. You come up with a new idea, you need to pitch that idea, find funding, need to build a project and a team around that to get success. You have touched on. And a lot of people say in my young researchers say, oh, I'm overly skilled in this area, I'm not relevant to the world. And I think it's just not true. It's just that they've not had the exposure to understand that. And so, as well as the resource that the industry can bring, they can also bring mentoring to help open the eyes of young researchers about what they're doing in the lab, how it can be related, whether it be funding applications, whether it be communications with the public, the policy, the government. These are all important parts of an academic career that the industry have got a whole wealth of understanding and years of experience in. But really, it's about networks for me. The bottom line of getting anything done is people interactions. And just through this 1 month short course, I met people in business, I met engineers, high tech, just down the road on the M4 or in Bristol. And I've now got new collaborations providing hardware for my experiments. I've been doing some consultancy with medtech startups, for example, and getting paid for it. This is all through just having the exposure to meet new people. And this is really important. This is the sort of graph that surprised me. is that you can see it's a bit outdated from the Royal Society, but from those that start a PhD, only about 3%, 4% stay in the permanent research staff setting in academia. And still only 30% stay in research in total, rather being in on-university research like government or industry. And I think we can do better than that, in my mind. And a lot of the problem is that young researchers just fall off the end of the conveyor belt, not really knowing what to do next. Having these connections, having this mentoring early on is going to help either build better research to keep them in academia if that's what they want, or it's going to help them bridge and then take jobs in non-academic research settings. And of course, if they don't want either, they want to go into careers outside of science, just revealing to them the skill set that they've developed in those 6 to 10 years of research is going to help that. So I'm going to come to the end now. I can see about 15 minutes. We're going to have some Q&A sessions. So I guess what does this model look like? How can we get this to happen? And I think it's really about providing the environment and the opportunity for academic young researchers to spend time in incubators, in biotech startups in their local environment, whether that be in and around London or up north and around Liverpool and Manchester, and investing in those networks of interactions. And I would really call on the UKRI to do a bit more in this space. I know they want to and they're trying to, but they have the grease really for the wheels of this. They fund the research, they pay for the salaries of the postdocs, they know the industry connections. And I'm trying to create a way of making that happen more often, I think would help UK science in general, but also, like I said, shape that future for drug discovery. So I'll finish there. I hope I got my point across. Thank you for listening. If anyone wanted to contact me and discuss these ideas, I'd be very willing to do so. You can get me on Twitter or my email address.
Thank you very much to both our speakers for their informative and encouraging talks. In the interest of time, I would like to go through questions that have been posted. The first question is from Tom Blackmore, and I think this goes to John. Is there a resource available anywhere that describes all of the industry or academia grants or partnerships available?
Actually, Robbie might be better at answering that than me. You mean when you want to actually find a source of funding? Not to my knowledge. I mean, a lot of them also have been built between particular companies and particular universities, for example. So some of the companies active in the UK, like BMS or UCB, they run fellowships schemes in conjunction with different universities. Similarly, the Evotech incubators, there's a number of those set up involving several different UK universities, but I think the only way to access them is through the portals within the university intranets. So my answer is no, I don't think there is one stop shop.
Thank you very much, John. Thank you.
So I've got the next question listed in the Q&A. This is from Isatau Sar. Quite a complex question to answer, so I'll put it to both of you. What can be done to overcome the kind of specific challenges that come from engaging or having a target you need to engage in a complex neuronal network? Obviously, there's difficulties quantifying these kind of engagements and also toxicity. Is there anything that can be done to overcome these challenges?
Do you want to have a go first, Robbie?
I'll have a little go. I guess, like you pointed out, it's complex. I think you've got to be thinking more targeted about what's important. What's the measurements that are going to allow you to translate across to the human without getting too bogged down in what goes up, what goes down, characterizing the whole network. There's many drugs that we use that we still don't really understand how they work. It's if they do work, can we get a feel on whether this is going to cause us problems? And if it's not, then the rest is potentially not necessary from an industry standpoint. From an academic curiosity standpoint, it certainly is, but I don't know if that's a useful response.
Thank you very much. Sorry, John.
Yeah, there were several things in your question, unfortunately. From the point of view of studying neural pathways, then I think it comes back to the fundamental. What you want is a pharmacodynamic assay. That's more important than a disease model. I think it's the disease models that have led us up the garden path. If you can focus in on something that has a smaller black box, say I want to understand the drug effect on, say, the excitability of this particular class of neurons, then you can use your animal models to study that, using in vivo electrophysiology, for example. And I think that's how you get around those problems. Toxicity, well, we actually conduct huge panel screens to try and weed out CNS toxicity. And all of the panel screens, like SERP, contain a lot of the neurotransmitter or CNS active GPCRs, ion channels, etc., so that we can have a fair stab of starting off with a molecule without obvious toxic side effects. And in fact, in my experience, in my career, I've had more problems with molecules not doing anything at all and there being no apparent CNS side effect. And it would have been nice to have seen a little CNS side effect to know it was getting into the brain properly, rather than massive side effects. So I think, tox is something that we've had to grapple with for years and years and years, and the toxicologists are pretty good at it.
Thank you very much. The third question is from Izato Sar, asking, the limited translational value of animal models used in drug discovery research has impacted in the drug discovery pipeline. Is there a trade-off between mathematical models or algorithms and biological models to overcome this hurdle?
Well, I think AI and computational analysis is making headway. Robbie actually touched on a really nice example that I wasn't familiar with in terms of the in-depth analysis of motor behavior in the pain models. And you can actually see that in all elements of biological scrutiny. So, for example, machine learning analysis of images from high content images is providing incredible depth. And it's just so much more comprehensive than saying use one marker. So I think there is a balance there. Using these systems will improve our critical assays. But at the end of the day, you've got to train them on something. And I think that's where you come back to using a whole animal system, as wide a physiological box as possible. Reveal stuff that you'll never have thought of training your model with.
I think just to add on the end there, John, your point about being human-centric, having access to patient experiences and clinical notes, training them on a model that's modeling the wrong thing isn't going to help anybody. And thinking about revisiting these models in psychiatry with the influence of a new age of thinking, putting the human condition first, it needs to happen. These things need to change.
Yeah, it's a good point. And I think, as you say, being smarter about these things, we can kind of ascribe the valley of death of translation to our limitations of perhaps some classical models of psychiatric disease in animals, but being smarter and applying these kind of new ways of analyzing things and refining these models is probably the way forward. That might be the balance. I've got another question from Peter McCormick, and that's a specific question for you, John. At DDI, do you make your own molecules in the medchem and pharmacology step, or is that an in silico ordering process?
No, we do make our own molecules. either in-house. It's about 50% in-house with our own synthetic chemists, 50% outsourced. But you rapidly come up against the buffers if you just try and buy and use already available molecules. So ultimately, you want to be making your own molecules so that you can get some novelty. Otherwise, your chemistry program won't go anywhere.
Thank you. The next question is from Kevin Cox, who is directed to John. Kevin says, "Hi, John, an amazing case study for getting a target adopted. Clearly, the ability to connect half a dozen different organizations was key, no doubt helped by your combined academic and industry career. However, how can an inexperienced academic, albeit with great science, begin to identify and build such a consortium?"
That's a good question, but I think you'll probably be surprised at the resources available to you. You know, you could consult colleagues in the Psychiatry Consortium if you're working in this field. Also, your IP groups and tech transfer officers in the university will have a lot of contacts, possibly as a result of spinning out companies, for example. So they're always worth going to. And because universities have got to raise revenue from a diverse range of sources now, they have been growing their alliance groups, and they're spending a lot of time courting pharma companies and industry. So they will have places you can go to. And in Oxford, we also have entrepreneurs in residence whose job it is to advise academics. And if you don't find them in your own university, go and look for them in another university, because these guys actually don't have much allegiance to one university or the other and could work all the way across the UK higher education spectrum.
Thank you very much, John. That was a good answer.
Great.
Yeah, great.
So there's no more questions in the Q&A. If you have one, quickly type it up. We've got a couple of minutes left. John, I was thinking about you had a kind of free approaches, the free way to skin a cat. I guess that's more of a ways to translate your research to impact as an academic, DIY, incubators and pharma collaborations. Do you have any idea of what the success rates of these kind of approaches have looked like in the past?
Well, they're all going to vary a lot, of course. I think, I mean, we're pretty successful here in terms of industrial collaborations. You know, we've more than raised the same amount of academic funding, again, through industrial collaborations. The incubators, they have a different type of success rate. So Lab 282 is one of our incubators here. It has funded over the course of 3 years, something like 38 different projects. So I suspect that is slightly less than 50% success rate at getting funding, but I can promise you it's way above your grant success rate would be. And that's what I mean by their low energy. As these, and the application forms are trivial compared to a Wellcome Trust Collaborator Award or such like. I mean, they really are. You could fill it out in an afternoon by cutting and pasting from a previous grant application. So I think the success rate is really good, but of course you've got to be interested in doing drug discovery because they've got a narrow niche, these incubators.
I think we just have one last question. Sam, if you would like to read that off, that's fine.
Yes.
Yeah.
Have we? Yes.
Right.
Sorry, one's just disappeared, but there's one remaining. So an anonymous attendee asks, how can academics without any connections make small contributions? I guess that's
well, Robbie gave lots of examples of this. you know, I sit here in the university trying to exploit advances made by academics, and they are all shapes and sizes. An assay, a cell line that's been well characterized, a novel target from an siRNA screen, or even just further data to validate and corroborate a hit from a screen like that, using, you know, primary cell models, either from animal cells, perhaps, or from patients. It's legion where you can contribute. And I think, as one of the previous questions said, you know, maybe you've got one part of this, how do you find other people to stick it together with? Just find other people who are sharing your passion for a particular mechanistic area. You'll come up with something.
That's great. Thank you very much. So I guess it's up to me to close the session. So thank you everyone for joining. Thank you to Robbie and John in particular for the really great and fascinating talks. Thanks to the BNA and the Psychiatric Consortium for supporting this session. And please join us again for the next part of this webinar series. It's on March 8th, I believe, and that's for an industrial perspective on drug discovery. So thank you very much.
Yeah, and thank you so much from us at the BNA. Thank you to everyone. And if anyone would like any further info on this or any of the other BNA projects, then please do contact me directly on my email, which will be in the chat. And finally, a little BNA plug that for the price of a coffee a week or less, you can help support events like these and keep them as accessible as possible. And we'll put a link in the chat there. And we look forward to seeing you on the 8th of March. Have a good day.
Thank you.
Thank you, everyone.